Hi everyone, my name is Paige and thank you so much for joining me for our lesson on the oxidative burst. It's also known as the respiratory burst, and it's actually an immunology topic, but there's just enough biochemistry sprinkled in there that they let me do it. I think this is one of the coolest topics. So we're going to be looking at phagocytes, and specifically what's happening inside of a phagolysosome. When a phagocyte has captured a bacteria, it has to find a way to kill it. And I just finished telling you all about the dangers of reactive oxygen species in the last video on the pentose phosphate pathway, but your phagocytes are going to just spit in the face of danger. They're actually going to create reactive oxygen species, and in doing so, they're going to create a biological weapon to aim at and kill and destroy these bacteria. So the first part of this burst is going to be to create reactive oxygen species within a phagocyte to help the immune response. The second part of that then is, now that we've unleashed the beast, those reactive oxygen species, we are actually going to have to neutralize them to make sure that they only kill the bacteria and don't kill your cell in the process. And so that NADPH that we just finished creating in the pentose phosphate pathway is going to be critical for both halves of this, right? Both the production of the reactive oxygen species and then the neutralization when we're done. So this in blue out here, right, is our phagocyte, and it is phagocytosing a bacteria. And so this sort of bleb in here is the phagolysosome that is basically gobbling up and eating the bacteria. And so we're going to start off looking inside the phagolysosome as to how that killing actually occurs and that generation of the reactive oxygen species. Big shocker to you, I'm sure, is that in order to generate reactive oxygen species, we have to use oxygen. We're going to use enzyme 1 right here, which is called NADPH oxidase, and we are going to use the NADPH that we made in the pentose phosphate pathway to turn that oxygen into a free radical called superoxide, which is oxygen with an unpaired electron. Then we're going to take enzyme 2 called superoxide dismutase. We're going to take that superoxide and turn it into peroxide. You can see now we've sort of reached a fork in the road here with what we can do with peroxide. We're going to continue on down the path for now, and we'll come back to those side paths in a minute. But right now we're focused on our end goal of just killing the bacteria. So we're going to take enzyme 3 here called myeloperoxidase, and we are going to turn that peroxide, combine it with a chloride to turn it into hypochlorite, which is HOCl, again with an unpaired electron. Now, interestingly, some invading bacteria can actually produce their own hydrogen peroxide. And when those bacteria release that inside the phagolysosome, they're just going to enter the pathway right here and continue on down the line. So your phagolysosome is equipped to handle that peroxide being made by these combative bacteria. And finally, we're at the part where we get to kill the bacteria. We're going to take that hypochlorite with its dangerous unpaired free electron, and we are going to kill the bacteria, split it right open. Now, let's go back to peroxide and talk about these two other branches here. I just finished telling you that invading bacteria produce their own hydrogen peroxide, and that your phagolysosome can harness this in addition to this whole oxidative burst pathway in order to kill the bacteria. However, some bacteria have an enzyme called catalase. This is a property of the bacteria, right? This is not intrinsic to your phagolysosome. The bacteria have this with them, and it makes them more resilient because they're able to neutralize their own hydrogen peroxide into water and oxygen. And so this means that your phagolysosome cannot turn your bacteria's hydrogen peroxide that they made back against them. They're able to neutralize that. And that'll be important in some pathology we'll talk about at the end of the video. All right, so we did it. We killed our bacteria. Fantastic. Now we have these reactive oxygen species floating around in the phagolysosome, and like I said, we can't let it stay there or it's going to destroy the phagocyte. So that hydrogen peroxide is going to come into the phagocyte, and that phagocyte is going to use an enzyme we talked about when we talked about the pentose phosphate pathway last video, this enzyme 4 here, which is glutathione peroxidase. So remember, reduced glutathione was a really special product that could detoxify peroxide. When you take that reduced glutathione, combine it with the peroxide, you can do a redox reaction. In the process, you will form oxidized glutathione, and that peroxide will turn into water. So when I was first learning this content, I did not understand the difference between catalase and peroxidase. Both of them are taking peroxide and turning them into something harmless. Broadly, the difference between a catalase and a peroxidase is that catalase is going to use 2 peroxides, and it's going to just sort of shuffle the oxygens around and form water and O2. A peroxidase is going to take 1 peroxide and then another source of reducing power and make different products. So instead of 2 peroxides, it's 1 peroxide and 1 something else. In this case, the something else is reduced glutathione. And we're still forming water, but then our other product is going to be the reformation of the oxidized glutathione. So we just talked about peroxidase, which formed water. But we had to sacrifice a reduced glutathione, which became oxidized glutathione. And so we want to find a way to reform that reduced glutathione so we can go on and detoxify more peroxide. Remember, we reform reduced glutathione by using the reducing power from NADPH. The enzyme that does that is enzyme 5 right here, glutathione reductase. And all that step 6 here is showing you is just that G6PD step, right? That first rate-limiting step of the pentose phosphate pathway. That was the first NADPH producing step of that pentose phosphate pathway. So that's where the NADPH came from. So our neutralization reaction is happening at this step here with enzyme 4, glutathione peroxidase. Steps 5 and 6 here are just showing how you regenerate that reduced glutathione, right, via NADPH, and where that NADPH is coming from. It's reminding you how all of this ties in to the pentose phosphate pathway, which is in this purple box right here. All right, take a breath. Let's recap on the outline here. Remember, the first step of the oxidative burst was turning O2 oxygen into superoxide, which was that oxygen with an unpaired electron. The enzyme that did that was NADPH oxidase, and that required NADPH from the pentose phosphate pathway. We took that superoxide and turned it into peroxide via the enzyme superoxide dismutase. We combined that peroxide with a chloride, and we formed hypochlorite, and the enzyme that did that was myeloperoxidase. Now, this myeloperoxidase enzyme contains heme. It also has a blue-green pigment, and that is why when you cough up sputum when you're sick, it's the myeloperoxidase enzyme that gives the sputum its, blue-green color. And then some side points we made about peroxide: invading bacteria could generate peroxide by themselves, and it would just sort of enter the pathway here at this step, and the phagolysosome could take that peroxide and turn it against the bacteria. But those extra sturdy bacteria that have that enzyme catalase, we call those catalase positive organisms, they had that bacterial catalase enzyme that could neutralize the peroxide into water and oxygen. So your body isn't able to turn their own peroxide against them. And then in that last step, the hypochlorite, which is basically bleach is what hypochlorite is going to go and kill that bacteria, and it's going to promote the release of lysosomal enzymes. And now the phagocyte has to find a way to neutralize all that extra peroxide. It's going to do that by using the enzyme glutathione peroxidase to combine that peroxide with reduced glutathione to turn that peroxide into water and then oxidize the glutathione. And so at this point, we've successfully carried out our neutralization reaction, but we need to remake that reduced glutathione for the next neutralization reaction. That's what these next 2 steps were about. We reformed that reduced glutathione by that enzyme glutathione reductase, and we reduced it with the reducing power from NADPH from the pentose phosphate pathway. And one of those steps in the pentose phosphate pathway that we were interested in was that G6PD rate limiting step. And like we talked about in the last video, if you're deficient in G6PD, you're not going to be as robust against oxidative stress because you're not able to neutralize those reactive oxygen species as efficiently because you can't reduce the glutathione to participate in that detox reaction.